So thank you for the introduction. And today I'm going to be highlighting some results from our co-occurrence of pesticide and PFAS study in California ag streams. So this study came to be after we read a Guardian article that was released in May of 2023. In this article, several pesticide products were identified with links to PFAS. And after reading that, we decided to design this study around one of those products, Intrepid 2F, which is the second most widely applied pesticide in California after Roundup. The active ingredient in Intrepid 2F is methoxyfenazide, so that was one of the compounds that we were really targeting in our pilot study design. Digging into the literature, there are other studies that have looked at pesticide and PFAS links. So there's a 2022 study that looked at various insecticide formulations for target PFAS as well as precursor compounds. Precursor compounds are compounds that oxidize in the environment to our target PFAS, or what we call terminal PFAS. And then more recently, there was an article in Environmental Health Perspectives that identified multiple pesticide products with links to PFAS. The Intrepid 2F product was named, which was our focus one from the Guardian article, as well as several other pesticide products. In this article, they talked about various sources of PFAS. So our traditional line of thinking was that the PFAS were coming from the fluorinated containers, but this article also suggested potential sources such as the active ingredients and inert ingredients, and then adjuvants, solvents, and potentially manufacturing byproducts. So we chose our sites across California to capture methoxyfenazide inputs. So the figure on the right, it's a little bit hard to see, but the lighter yellow colors are low methoxyfenazide usage and the red colors are high methoxyfenazide usage. So we chose 5 sites in the Sacramento Valley and 5 sites in the San Joaquin Valley that were around these high methoxyfenazide application points. And another thing with our site selection was that we were really trying to isolate ag inputs and minimize urban inputs. So while our sites were chosen based on methoxyfenazide, we likely captured other pesticide products because we were capturing various crop types. So rice, nuts, tomatoes, alfalfa, and row crops. Our sites represent surface water streams as well as several ag drainages. And during the time of sampling, we saw very little rainfall, which I'll touch on in a following slide. So our site samples were likely influenced heavily by irrigation and return waters. Some photos of our site below, we see Ingram Creek dominated by almond crops, the Reclamation Z drain dominated by pasture and alfalfa, and then our Colusa Basin drain site dominated by rice. And then in terms of sampling timeframe, again, we were really trying to capture Intrepid 2F and methoxyfenazide. And the great thing about California is the California Department of Pesticide Regulation tracks pesticide use data across the state. So we were able to pool methoxyfenazide use data from 2021, which was the most recent year we had available at that time, and plot that. And you can see there's 2 light tan bars that are highlighting the high use periods from late April to early May and late June to mid-July. So we targeted our sampling around May and July during those high methoxyfenazide use periods. One thing I wanted to point out, at one of our sites, we did see some aerial applications taking place during sampling when we sampled Snake River in July. This might explain some results we're going to see later, but maybe not. It's just something we wanted to consider when we're interpreting results. So in terms of surface water sampling, we followed USGS protocols. We used a weighted bottle sampler. We do a clean hands, dirty hands procedure, especially for PFAS. And it's important to point out that the USGS has tested all of the materials that were used for PFAS to ensure that we're not contaminating our sample with, say, a certain type of plastic container. We collected 2 mil polypropylene tubes half filled for our PFAS analysis, and then 1 liter amber glass bottles for the pesticide analysis. We did do 2 replicates and 2 field blanks as part of the study as well. So for analyses, our pesticides were analyzed at the Organic Chemistry Research Lab in Sacramento, California, following a published USGS method that's linked on this slide. It's 180 plus compounds, and that pesticide analysis goes through GCM-SMS and LCM-SMS. For PFAS, we did a target analysis at the National Water Quality Laboratory. It's around 60 compounds total, and that was run on an LCM-SMS. So I wanted to talk about the hydrology around the time of sampling. I think this is really important to point out especially when we go to do interpretation. So at our Sacramento Valley sites in the north, there was no precipitation the 24 hours leading up to our May sampling, and no precipitation the 24 hours leading up to our July sampling. In the San Joaquin Valley sites in the south, there was some precipitation before the main sampling, which is highlighted in the blue table, but you can see for all sites, it's less than an inch per site. And we had very little rainfall leading up to the sampling, so likely this small amount, this less than an inch, went into the soil and there was very little runoff actually occurring. And then in July, there was no precipitation before sampling in the San Joaquin Valley sites. And the reason I want to point this out is, again, what we're seeing with our results are probably more influenced by irrigation and ag return waters than, say, rainfall runoff. And to further drive that point home, this is a hydrograph of Del Porto Creek upstream of the site we sampled along Del Porto Creek. But you can see there's a gray bar there that's highlighting our sampling window, and we are operating under these base flow conditions. So again, base flow, little rainfall, little runoff, a lot of irrigation, ag return water influencing our results. So the next 4 to 5 slides are going to highlight some of the pesticide results. So again, we analyzed these surface waters for, I said 180 plus, it's exactly 190 compounds. We detected 69 unique compounds across all sites and samples, and 3 compounds in particular had 100% detection frequency: azoxystrobin, which is a fungicide, tebuconazole, also a fungicide, and then methoxyfenicide, an insecticide, which is encouraging because that was that compound that we were really trying to target with our sampling. The maximum concentrations for all 3 of these compounds occurred at Mustang Creek, and that's also where we saw the highest concentration in the study, 782 nanograms per liter for fluxapyroxide. And Mustang Creek is in the San Joaquin Valley. I circled it in red on the map. And then there are some photos highlighting this site, and you can see that it's really like an ag drainage ditch. So we decided to break the pesticide results down by class into fungicide, herbicide, and insecticide. We detected 20 unique fungicides, 28 unique herbicides, and 18 unique insecticides. Now the sites are arranged from left to right. The northern sites on the left, the Sacramento Valley sites, and then the San Joaquin Valley sites are on the right. So those northern sites are highlighted in a light blue, and the San Joaquin Valley sites are highlighted in a light green. And you can see that there are some differences between the 2 valleys. So the Sacramento Valley sites have a little bit more dominance by the herbicides versus the San Joaquin Valley sites have a little more dominance by the fungicides. And this is likely relating to crop type. So those Sacramento Valley sites have a lot of rice, versus the San Joaquin Valley sites are a mix or dominated by nuts. So that's something we're going to explore further as we work through the data analysis. So the next slide I want to highlight are methoxyfenicide results. So again, this was our target compound when we designed the study. So methoxyfenicide was detected across all sites, 100% detection frequency, and our highest detections occurred at Mustang Creek, the high concentration of 497 nanograms per liter. But one thing I wanted to point out is that high methoxyfenicide concentration at Snake River in July. And if we think back to that previous slide where we did see aerial application occurring during sampling, there may be some link there, but that's something we're going to have to explore further. And then again, just to bring this back to PFAS, so methoxyfenicide is the active ingredient in Intrepid 2F. And in several papers and that Guardian article, Intrepid 2F has been linked to the PFAS, perfluorobutane sulfonic acid, or what we call PFBS. So it'll be really interesting to look at the PFAS results and see if we're detecting PFBS in these samples. So now I want to talk about the OECD definition of PFAS. So it's defined as a fluorinated substance that contains at least one fully fluorinated methyl or methylene carbon atom, and there are a few exceptions to this definition. So far we've been thinking about PFAS from kind of our target PFAS that are in our EPA's what we call 1633 methods, but this definition opens up an interpretation around pesticides that are considered PFAS. So I've identified a couple pesticides in the bottom right that meet the OECD definition of PFAS, and you can see I've circled the fully fluorinated methyl groups on those compounds. And then on the left, I've identified a few of our PFAS that are in our target methods, just so you could see how these structures compare to the pesticide structures. So if we look at our pesticide results in the context of the OECD definition, we had 35 compounds that met that definition, and 14 of them were detected. And I've listed those 14 compounds here. The most frequently detected pesticide was flupirum in 16 of 20 samples. And again, the highest concentration was at Mustang Creek, which we've been seeing for a lot of our results. Flupirum is a fungicide that's applied to a wide variety of crops. So to put some of our results in the context of aquatic life benchmarks, we decided to plot up the compounds that were detected that exceeded benchmarks. So we have 4 compounds: bifenthrin, cyhalothrin, diuron, and imidacloprid. And bifenthrin is one of the fluorinated pesticides that meets the OECD definition of PFAS. So that's an interesting one to point out. And these 4 compounds exceeded various acute and chronic benchmarks for invertebrates and vertebrates. Once we get the PFAS results, we will be putting those in the context of recently released EPA benchmarks, and they came out in September of 2024. So some other organofluorine considerations. So something that we want to think about moving forward is total organofluorine, which will be an indicator of all of the fluorinated organic content in a sample, whether that's from pesticides, PFAS, or other compounds in the environment. And then something called trifluoroacetic acid, which we may be able to go back and run on some of our samples. We're considering that moving forward. This is an ultra short chain PFAS. It's mobile and persistent. It's been detected in various matrices and is considered a byproduct of some of these fluorinated pesticides and other fluorinated contaminants. So unfortunately, I do not have PFAS results to share. I was really hoping to get those before this talk, but the analyst is still working through them. But we did run 2 methods on PFAS. We did a direct aqueous injection method and a solid phase extraction method. The online SP method has lower reporting levels and analyzes 30 more compounds, so that's where we get that 60 total compounds. As soon as I get these results, I'd be happy to share them with the audience. They'll be published in our database for the public. And then we're also planning to write up a journal article sharing these with the scientific community. So to bring this back to the Chesapeake Bay, thus far, we've really been focusing on California, but this work does relate to work that's ongoing in the Chesapeake Bay. So Samuel Miller at USGS released a paper in 2024 looking at pesticides in the Potomac River watershed. And what I found interesting was that some of the pesticides that are detected in the Potomac are pesticides that we're detecting in California, which would suggest that similar products are being applied. Methoxyfenazide and pyperonal butoxide in particular were detected in this Potomac study, which are active ingredients linked to products that have been linked to PFAS. So again, methoxyfenazide was our target compound in California, and that was something being detected in this Potomac study, albeit at lower concentrations than what we detected in California. And then I've also highlighted in red here that some of the pesticides that were detected in the Potomac are pesticides that meet the OECD definition of PFAS. So those are also showing up in the Chesapeake Bay. Another group at USGS is looking at land cover and what they're calling different typologies across the Chesapeake Bay. So pasture, mixed ag, row crop, and developed land use. And they're looking at surface water samples and doing target PFAS and select pesticides for those surface waters. And that work is ongoing. The data analysis on this will start in the spring, and that will be something very relevant to the Chesapeake around pesticide applications and PFAS. And then finally, there's another group at USGS that's looking at biosolid applications in the Chesapeake. They put out a USGS data release identifying biosolid inputs in Maryland, West Virginia, and Delaware. And there are questions around these biosolid inputs and their relation to PFAS. So that's something that we will be exploring in the Chesapeake moving forward. So in summary, we can say that pesticide use may represent a source of PFAS in ag drainages. Fluorine-containing pesticides are also present in ag drainages. And in general, pesticide applications and ag releases to surface waters do warrant further study. So with that, I just want to thank the USGS Environmental Health Program for their funding of this pilot work, the Organic Chemistry Research Lab that did the pesticide analyses, and my co-authors. And I'm happy to take any questions with the time that's remaining. Thank you.